Alrighty guys, here we are again. Had to stop it because it was getting too, oops, not from the start. Nope. Had to stop it because we were getting down there and yeah, towards 30 minutes, and sometimes YouTube gets upset if my videos are too long. Alrighty, so special cases, glycine, proline, and our last one is cysteine. So cysteine, if you were looking at this in the chart, you'd see that cysteine has a sulfur, an S. So this is very special because these sulfurs really like to bind with themselves. So sulfurs and sulfurs like to bind together. And so these can form what are called disulfide bridges with other cysteine amino acids. And this is not just any old bond. This is a strong covalent bond. So sharing of electrons and all that kind of business. This can reinforce the folding of the protein, and all that will make more sense in a few minutes. But essentially, if you've got, a string of amino acids, it's very, very long, maybe all their stuff's going on over here, oops. And then you've got, the string continued on. So even though these cysteines might not be close together in this amino acid sequence, they can form this double bond together. Alright, so again, your 20 amino acids, whenever I ask questions about these things, you will always have this chart or pictures of them or whatever. Alright, when we want to make a polymer out of amino acids, which we would call a polypeptide, and after it's all done and ready to go, we call it a protein. Oh, guess what kind of reaction it's going to be, guys? I bet you got that right. It's a condensation reaction. So we are releasing some H2O here, and we form a bond between amino acids called a peptide bond. And we can do that again, and again, and again, and again, and end up with a really, really, really long string of amino acids. So amino acids, again, they're linked by a condensation reaction, and that bond is called a peptide bond. The sequence of amino acids is what we call the primary structure of a protein. Okay, and here's where things get cool and wild. So we've got 20 different amino acids, right? So how many different potential combinations of, just two of these are there? How many different polypeptides can you make that are only 2 amino acids long? Well, to figure that out, you multiply the number of amino acids by the number of amino acids again, because you can't have it bounded with itself. That's why it's not 20 to the 19. So you multiply 20 times 20, and you can figure out that there are 400 possible dipeptides. If you just want something that is 3 amino acids long, there's 8,000 possible tripeptides. If you want to have a polypeptide that's 100 amino acids long, this would actually be a very small polypeptide. Most proteins are made of way more amino acids than that. But if you did that, it'd be 20 to the 100th power possible polypeptides that are 100 amino acids long. We're not even counting all the ones that are 2 amino acids long, 3 amino acids long, 4 amino acids long, etc. Only 100 amino acid long ones. 20 to the 100th power. That is basically such a giant number that I cannot even write it. Okay, and you may not believe me here, but I am telling you guys that that is more than the number of electrons in the universe. Think of all the atoms with all the electrons. Alright, mind blown, right? So we can take these 20 amino acids and we can string them together in different ways, like beads on a string. And we can make almost literally infinite combinations of those things. So that's pretty cool, right? Like, that's pretty amazing, I think. And it's important what the sequence is because the amino acids in different parts of the polypeptide chain can interact with one another. And this is where we get other levels of structure. Okay, so primary structure, just the sequence of amino acids. Secondary structure, you can have either alpha helices or beta pleated sheets. So in an alpha helix, just like a DNA double helix, but this is a single helix, you have hydrogen bonding between amino acids, and it makes the whole thing kind of wind up around itself. In beta pleated sheets, you'll have some amino acids going this way, and then booping around over here, there's a whole string going on, and then it boops back over this way, and you have pretty distant parts of the whole polypeptide interacting with each other, hydrogen bonding. And instead of making this alpha helix structure, it makes this kind of like weird pleated structure. So that's a beta pleated sheet. Okay, so primary structure is just a sequence of amino acids. So which R groups do we have going on? Secondary structure is how they start to interact with each other in alpha helices or beta pleated sheets. And then we have tertiary structure. So it's starting to get real crazy. And this is where that stuff folds up even more. And if that's not enough, we have quaternary structure. So that's where different chains of amino acids, so different polypeptides, have to come together to form a functional protein. This is hemoglobin. So hemoglobin has quaternary structure. Not all proteins have quaternary structure, but hemoglobin does. So hemoglobin has these 4 subunits that come together, and then they carry an iron right there in the middle. And the iron helps our hemoglobin carry oxygen. Okay, so protein structure. Make sure you guys can identify these things. Make sure you know what an alpha helix looks like and a beta pleated sheet. Make sure that you know the different levels of structure of polypeptides. All right, so again, the amino acids in different parts of the chain interact with each other. And that makes the secondary, tertiary, and quaternary structure. You can have a bunch of different sorts of interactions. So you can have hydrophilic interactions, because, you know, charged and polar stuff isn't just attracted to water. Charged and polar things are attracted to other charged and polar things as long as they have the opposite charge. Okay? So the different R groups can be attracted to each other as well. You can have hydrophobic interactions. That's where different hydrophobic parts, or, you know, hydrophobic R groups, are attracted to each other. And in cells, now this is key for your homework worksheet thingy, in cells a lot of the time this sort of translates into what part of the polypeptide is all mushed in towards the inside, and what part is sticking out towards sort of the outside part of the crumpled up paper that I think of as a polypeptide in a cell, and having to interact with water. Okay. Of course, the structures can have hydrogen bonds between them, or you can have disulfide bridges. And which amino acid was that can make these special disulfide bridges between two of themselves? Cysteine, yes. Cysteine has a sulfur. They can covalently bond with one another. Very cool. All those interactions, again, cause the folding of the macromolecule. So secondary structure, alpha helices and beta pleated sheets. Okay, that's the alpha helix, beta pleated sheet. Then you have tertiary structure, and sometimes quaternary structure determined by the R groups. By the way, you might see proteins, looking like this. This is what we call a space filling model. And it shows, this is kind of like a little bit more realistic version. Like if it was possible to have microscopes that can see this stuff at the molecular level like this, literally at atomic level, then this is kind of what they would look like. This is just sort of a graphical tool we use to draw these things out so that we know what the structure is doing. But it would literally look like a balled up wad. All right, so that is actually hemoglobin. The different colors are the different subunits. Okay, so now you should be able to think about this question, and this is on that worksheet. So what is it that determines which amino acids point inward versus outwards? Think about the environment these proteins are in. I just gave you guys a clue a second ago. And think about this as a watered up sort of piece of papery thing, kind of like that space filling model of hemoglobin. Think about the properties of the R groups of the different amino acids. Okay? I'm not going to tell you guys the answer to this one. I want to see you guys do it. So what determines which amino acids point inwards versus outwards? We'll talk about this on Tuesday. All right, so now we have explained the functions of enzymes. And, this next and last learning objective for this chapter we will be accomplishing on Tuesday. So, you guys are all done with your assigned video lecture for Thursday. And make sure that you guys are studying hard. Please, please, please come talk to me, come see me. Even if you just need study tips, talk to the tutors. Use the Center for Academic Success. Use your time at study table wisely. Actually study instead of just socializing. Find a partner in the group. And just, more than anything else, make sure that you guys are already studying. Just study for that exam so you're not cramming everything into your brain at once. It has nothing to do with how smart you are. It's just that our brains literally have a limit on how much they can memorize on any given day, any given time period. Okay? So stretch it out. Do yourself a favor. All right. Have a wonderful day, everybody, and I hope that you are staying warm. Bye.